Good evening, everyone. I'll wait for 2 more minutes to see if more people are joining, then I'll start the session. Good evening, everyone. I welcome you all to week 7 of this NPTEL problem solving course for the course Cell Culture Technologies. So, if you have any doubts in between, in the middle when I'm explaining anything, you can just write it in the chat box. So, I'll begin the session now. The first question is, which of these can be used to separate population of two cell types from a mixture? So the first option is immunopanning, second is fluorescence assistant cell sorting, third is both A and B, and the fourth option is none of the above. So if anyone has any idea of which of the following options is correct, you can type it in the chat box and then I'll discuss. Yes, so the correct answer is both A and B. So basically, both of these techniques, immunopanning as well as FACS, so this fluorescence assisted cell sorting is also called FACS. Both of these can be used to separate two different kind of cell population. So I'll just discuss in brief both immunopanning and FACS. So what is immunopanning? So as the name suggests, immunopanning. So it's basically a kind of panning which is based on some immunological reaction, some antigen antibody reaction, right? So immunopanning is basically a technique which utilizes immunoprecipitation of cells. So it's immunoprecipitation of not a protein but of cells. So immunoprecipitation, what does immunoprecipitation mean? So immunoprecipitation is basically another name for immunoprecipitation is pull down. So suppose you have a protein or you have a mixture of protein and then you have your antibodies uh, and that antibody is specific to a particular kind of protein. So now from that mixture of protein, using that antibody, you can pull down that protein, right? Because that antibody is specific to that particular protein only. So in a similar way, this immunoprecipitation of cells can be done. Okay. So how is it done? Suppose a specific kind of antibody, if you look at this figure, then a specific kind of antibody is attached here to a solid surface, like it's the antibody is immobilized to a solid surface, for example a cell culture plate, and from there you can like pull down or immunoprecipitate your cells. So why immunoprecipitation? Because this specific antigen antibody reaction is involved. Okay. Now another, so here if you look at this diagram, so we have this mixture of cells, you can see green, blue, yellow, and we have these specific kind of antibodies. Okay, so these specific kind of antibodies are going to pull down only one specific kind of cells. Okay, now and then we can do a second like a second step of this immunopanning, where after one kind of uh, one kind of selection by an antibody has been done, a second round is also done by another antibody. So if you look at the selection after these particular selection, at the end you will see only one particular kind of cells, which you can see here are these red cells. Okay. Now an important thing to remember while immunoprecipitation of cells is that what happens is cells are larger than our protein complexes, like for which immunoprecipitation or pull down is used. So what happens is, if we use the traditional immunoprecipitation which is used for protein and antibody kind of reaction, what will happen? They may be damaged. So that is why what happens in immunopanning is only a single solid surface is coated with the specific antibody or the cell surface protein ligand. Okay, so this is immunopanning. Now the next technique is FACS. FACS or fluorescence assisted cell sorting. So there are uh, basically two different kinds of cell sortings which are used. One is FACS, which is fluorescence based. Another is MAX, which is magnetic based. So both can be used, but they have some differences. The basic difference is in the usage of fluorescence and in magnetism. Max is a little faster than fax, but fax is a traditional technique and it has been used, it is used widely for cell sorting. So this is basically another name is flow cytometry. Okay, so now what happens in FACS or flow cytometry is, so basically we have specific antibodies, as you can see here, these are antibodies. Okay, and by utilizing these specific antibodies, what happens is, using a FACS analysis, you can simultaneously connect data on what kind of samples, what kind of cells are there in your sample, and you can also sort different kinds of cells using different parameters, whether it is a specific marker on the cell surface, whether it is cell size, etc. So what happens is, so here in a liquid stream, so these are your cells, these green dots and your black dots, these are your cells. And in this liquid stream, what happens? It passes, so this liquid, through this liquid stream, this laser beam passes, as you can see. So what will happen now is that every time a cell will pass through this laser beam, there will be an interference in this laser beam, right? So now what are we utilizing is this interaction of this cell with the laser beam. If there is no cell, then what is happening is there will be no interference in this laser beam. So and so this is the basic principle of flow cytometry. If it is like attached, if the cell is attached to a specific kind of fluorophore, and so as soon as a laser interacts, there will be a difference in fluorescence intensity. Okay, so depending on that, the cell sorting can take place, as you can see here. Now, when the cell comes in the path of the laser beam, we can get different kind of scattering. One is forward scattering, one is side scattering. I'll discuss these later. And these two angles are very important. So both this forward scattering, both the data on this forward scattering and as well as the side scattering is taken into consideration, so that we can get an idea of what kind of cell is passing through. And this is the way through which like the cells are sorted. So this, as you can see, so this is the light path. Your cell is in the light path. Okay. Here also in the second image also. This is the first image. The second image. In the second image also, your this cell is in the light path. Okay. So I'll discuss the forward scatter first. Now, so these two parameters, forward light scatter for FSC and side light scatter or SSC. So these two happen as a result of this your fluorescence emission signal, right? So this, what is this forward scatter? So basically this forward scatter is that light which is going to be refracted by the cell in the forward direction. So as you can see, this is your cell and this light path, this is being refracted in the forward direction. All right. And another thing which you can see is this forward scattered light is traveling in the same direction as your incoming light path. Okay. So there will be a very little deviation from this light axis. So this will be your forward scattering. Now, forward scattering is used to determine the cell size. Okay. So whatever information will come from forward scattering will basically be your cell size or your particle size. The cells or the particles which are bigger in size, they will produce more forward scattering. So bigger cells will lead to more forward scattering, and similarly your smaller cells will lead to lower forward scattering. So this is one criteria. The second is your side scattering or side scattered light. Okay. Now, this side scattered light, as you can see, it's not going to travel in the same axis as your incoming light. Okay, it's going to be refracted to different paths, and it's deviating from the original path. So this is the side scattering. Now, side scattering results from the complexity of your cells. So all your cells, like here, as you can see, cells have different organelles, and this results in different kind of granularity of the cell and the composition complexity of different kinds of cells is going to vary, right? Like your your leukocytes are going to have different kind of composition, different kind of granularity. Your erythrocytes are going to have a different kind of composition. So on the basis of what is the composition of each type of cell, side scattering will take place. So now, using the knowledge of the side scattering angle or the side scattering, we can know what is the complexity of the cell. So since each kind of cell will have a different complexity, we can, using that information, we can know, okay, like, okay, this is this kind of cell. Okay. So now, next question. Which of these is a signature for all leukocyte groups? So there are four options: CD45 plus, CD45 minus, plus means presence of CD45, minus means absence of CD45, CD340 plus or CD340 minus. So these all are markers. If you know the answer, you can uh, write the answer in the chat box. Yes, so some people have answered. The correct answer is CD45 plus. So the CD45 is a marker for the leukocyte groups. So basically, what is the CD? Like, what is CD45? What does CD stand for? So this is basically cluster of differentiation. So this cluster of differentiation, these CD molecules are used for like different kind of CDs are present on different kind of cell surfaces. So these are used as markers for a specific kind of cell. Okay. So basically these CDs have different kinds of function, like for example in cell adhesion, etc. And there are different kind of CD, and they have different kinds of nomenclature, and it is by the usage of the knowledge of these specific cluster of differentiation sites on different kind of cells, which is mostly exploited in our flow cytometry applications. For example, if you look at this graph here, as you can see here, so this is a flow cytometry data, and as you can see here, we have RBCs here, then lymphocytes, then blasts, then monocytes, neutrophils, and eosinophils. And as you can see here, all these have occupied different regions of the graph. So here, I don't know if you can see here. Here we have our side scattering angle, SSC, and here we have our CD45 marker. Okay, so here on the x-axis. So now once you have got both of these information, once you have got the side scattering angle And once you have got whether the CD45 is present on whatever cell is passing through the light stream or not, then according to that, if we plot it on a graph, then like you are analyzing the SSC against the presence of CD45 expression. Okay, so from here you can get a particular graph or a particular information of what kind of cells are present in the mixture. And every time you'll have a if you have a sample where suppose your RBCs, neutrophils, eosinophils, and these kind of cells are present, every time, no matter how many times you're going to do the experiment, if you plot this kind of graph, side scattering against CD45 expression, then what will happen? You will get this particular kind of graph only. You are not going to get completely different graph every time. So this is how this, now you can appreciate that this kind of information can be used for sorting different kinds of cell. Like for example, in the blood you have all these kind of mixtures, right? You do not have individual cells everywhere. Now from there, if you want to isolate or sort individual cells, then this FACS data is going to help you in isolating different cells, because each of the cells are going to come in different fraction depending on their different kind of forward scattering, different kind of side scattering, etc. So moving to the first question, third question, and is a protein molecule that recognizes what on the cell surface? Please answer if you know the answer. Yes. So the correct answer is an antibody is a protein molecule that recognizes antigen on the cell surface. So if you look at this image, you can see here like this is the antibody and it has its variable light chain, variable heavy chain, and then the constant region and so on, the disulfide bonds. Okay, and now on this antigen, on this antibody, you are going to have specific antigen binding site. So this specific antigen binding site is going to recognize your antigen. Now your antigen can either be any of the particles or any of your bacteria, any component of the bacteria, it's LPS, some cell wall structure, or your virus, etc. And so this specific antigen binding site on the antibody will interact with the antigen and it will render it harmless. Okay, and so these specific sites on the antibody which are present on these variable regions are responsible for recognizing and binding to the antigen. So moving to the next question, in uh, like a specific region, there are two, what we have to tell the name of that region. There are two types of motor neurons. One are small in size, which are called what, and other are large in size, which are called what. So in the last session, I discussed about the different kind of motor neurons, right? So if you remember that, so these alpha and gamma motor neurons I discussed extensively in the last session. Now, where are these alpha and gamma neurons found? and like which one are smaller in size and which one are larger in size. So I'll answer. So in the ventral horn, like there are two types of motor neurons which are present, and these gamma are smaller in size and alpha are larger in size. So basically, this ventral horn and dorsal horn, these are the regions of spinal cord tissue. So this diagram is a cross section. Okay, it's a cross section of the spinal cord thoracic region. So basically, there are two sides, right? Dorsal and ventral. So ventral means towards the stomach, this ventral side, and this dorsal means towards the back. Okay. So, but in this human anatomy, what will happen? So, ventral side of the spinal cord will mean the anterior side, right? Because it's the forward side towards the stomach, and this dorsal will be towards the posterior side. All right. So, and here you can see the location of the white matter, the gray matter, etc. So here in the ventral horn, as you can see, there are two different kinds of motor neurons. So this ventral horn has different kind of neurons and specifically the motor neurons. So here we have the alpha motor neuron and the gamma motor neuron. So in the last lecture, I discussed about these extrafusion muscle fibers and intrafusion muscle fibers and which kind of motor neuron plays a role in what. So these alpha motor neurons interact with the extrafusion muscle fiber and these gamma motor neurons interact with the intrafusion muscle fiber and the spindle. Okay. So the next question, in the rat spinal cord at E14 stage, so this E14 is a embryonic stage. All right. So in the embryonic stage 14, only motor neurons express a particular marker. This information can be used to isolate these cells from mixed culture with other cells of the spinal cord, test electrical activity of these cells, digest these cells, or none of the above. So, like, so you have a clue in your question itself, and I just discussed previously about this. Slide not moving. I think they are moving, right? You can see them moving. So yeah, the correct answer is A. So at any embryonic stage or at any stage, so it's written that these motor neurons are expressing a particular marker. So since they are expressing a particular marker, this information itself can be used to isolate these cells from mixed culture. And what will be the technique which can be used to exploit this information about this particular marker? So we can use facts to we can use facts because it is expressing a particular marker. So we can sort the cells, our cells of interest, depending on the usage of that particular marker, just as I discussed for the CD45 positive cells, for example, our leukocytes. So this is the same principle. Now, so this E which is written and this number which is written whenever we are referring to rat development or rat embryo development. So post-coitum, like post-coitum, what happens is we have different kinds of embryonic stages. So these embryonic stages, these are referred in the terms of day. So this is embryonic day 11.5, then we have embryonic day 12 and so on, and embryonic day 14, what we were discussing in the question before. So at different embryonic stages, different kinds of structures and different kinds of development happens. So that is the basis, on the basis of different kinds of structures developed, we have different kinds of embryonic stages. So this E14 which was mentioned in the question refers to that. All right. So I'll just discuss in brief how can we do motor neuron culture from a uh, from a rat embryo. So here, suppose, so the most important thing is when you are preparing your plates or your petri dishes, which I discussed, I think, in the initial lectures. So there, you have to coat your surface with some kind of ECM or some kind of molecule which is going to help the adhesion of your cells, right? So here, suppose we are coating our plates or our dishes with poly D lysine or laminin or any other kind of ECM which we have already discussed in the previous lectures. Okay. Then once you have coated your plates, then your plates are ready, right, for culturing. So then on the next day, what you can do, you can isolate the spinal cord tissue at E14 or E15 stage, which is the embryonic day stage. All right. So once you isolate that, you can once you get that, then you can make your single cell suspension, which also I talked about in the previous lecture. So by like dicing your tissue and then preparing a mixture, then centrifuging and so on. And from that, once you get like your spinal cord sample and once you prepare your single cell mixture, so after that, now what you have to do, you have to count the number of cells that are present in your solution. So for that, what we can do, we can count them using the hemocytometer, which also I discussed extensively in the last lecture. So once you have counted your cells, what now you can do? You can like add your cells, like you get your single cell suspension, right? And you also know what is in what volume of solution that you're taking, what is the number of cell because you have already counted, right? So once you have done that, now you can grow your cells, you can culture your cells. So that you can do using a specific medium in your already coated plates. Okay, and then you can go on culturing them. So this is the basic protocol of like isolating your rat neuronal cells and then culturing them. So moving to the next question, trypsin is used to like do what? Like what is the function of trypsin? By digesting the extracellular matrix as it is a first option. It is used to lift cultured cells off a surface. And is it a condensation enzyme? Or in the second option, is it a proteolytic enzyme? Then third option, is it used to attach culture cells off a surface? And again, is it a condensation enzyme or proteolytic enzyme? So if you know the answer, please reply in the chat box. Yeah, so the correct option is B. So trypsin is used to lift cultured cells off a surface as it is a proteolytic enzyme. So again, this is a basic flow of how to passage cells or subculture cells using trypsin. So the entire protocol of trypsinization of cells in order to passage them, I have already discussed in the previous lectures. You can go through these. So basically, trypsin is a proteolytic enzyme, and it digests the the bond which the cells have with the ECM or the coating on the Petri plates. And another important thing is that trypsin works at 37 degrees Celsius. So once we have our cells, we discard all the culture media, and then we add our trypsin, and after that we incubate the flask or the petri dish for 2-3 minutes, and then we wait for cells to detach, and then we look under the microscope that whether cells have detached or not. Once that is done, what we have to do, we have to remove the trypsin, or we have to neutralize the trypsin. So the serum which is present in our medium, cell culture medium, that neutralizes trypsin. And after that, once we know what is the amount of cells that we have, we can culture those cells again. So this is for what trypsin is used. And after trypsinization, so these are the cells before trypsinization, and this is the condition of the cells, like trypsin has been put in after some time, and then this is the properly correctly trypsinized cells. So this is how it looks like under the microscope. This figure here on the top, this is when it is trypsinized incorrectly. By incorrectly, I mean to say that, so a lot of trypsin has been added here, and that is why cells have detached in this clumped fashion. So this is the wrong way. Your cells should not look clumped once they get detached. They should look like this. They should look like single cells. Okay. So moving to the next question. Which of these is not a component of B27 supplement? Vitamin A, biotin, acutase, or sodium selenite? Please answer if you know the correct answer. Picture not clear. Which picture is not clear? This one. So I think it's some problem with the internet or something. A recording will be available, and I'll also share the slides. So from there you can access the presentation if it's not clear right now. So for this question, which of these is not a component of B27 supplement? The correct answer is acutase. So this B27 supplement is actually a neuronal cell supplement, and this is the composition. So here in the composition table, you can see that it has biotin, it has vitamin A, and it has the sodium selenite. What the other options that the questions mentioned, it doesn't have acutase. So this B27 supplement is basically used to support embryonic, uh, postnatal, or adult hippocampal and other neurons. And it is so in the previous lecture, if you remember, we talked about a medium which was the neurobasal medium. So this B supplement is used with this neurobasal medium to culture neuronal cells, hippocampal neuronal cells, and other, and also the other neuronal cells of the central nervous system. And the this component that the question mentioned, this acutase. So basically, this acutase is uh, is also somewhat something like trypsin, and it is used for cell detachment. What is acutase is different from trypsin in that it is not acquired from any animal. It is not mammalian. Its origin is not mammalian, and that is why it like in embryonic cultures, it is used so that we shift away from animal origin cultures or animal origin supplements. So as I mentioned, it is an alternative to trypsin, and unlike trypsin, which we need to neutralize, acutase doesn't need to be neutralized. Okay. So next question, glutamax is like what is the chemical composition of glutamax and has higher or lower stability than glutamine. So glutamax is basically this L-alanine L-glutamine, and it has higher stability than glutamine. So now, so this L-glutamine is used as a nitrogen source by cells, and so as a nitrogen source, it is used in the synthesis of proteins, nucleic acids, etc. All right. Now, a problem with L-glutamine is that once it decomposes into ammonia. Now, this ammonia is toxic to cells. So, this is a major problem in usage of L-glutamine in cell culture media. So, however, L-glutamine is essential, right? Because cells need a nitrogen source for protein and nucleic acid synthesis. So, this glutamax was developed as an alternative to L-glutamine. Okay, so basically, glutamax is this. It's L-alanine, L-glutamine. Now, so as you can see, it's a dipeptide, right? It has two, L-alanine, L-glutamine. So it's a dipeptide. So what happens in dipeptide is that this is the mechanism of dipeptide utilization. So it involves gradual release of this peptidase during culture, so that this dipeptide is gradually hydrolyzed, unlike this L-glutamine. So so here what happens? This is not This does not lead to cell toxicity. Okay, and this results in a high growth yield. So that is why this glutamax is used. So moving to the next question. Uh, this is a supplement used for serum-free fetal hippocampal neuron culture medium. B27 G5 collagen of ribroin. Yes, so the correct answer is B 27. So, what happens is that whenever we are growing neurons from the hippocampal cultures, they require different kind of factors, right? So, neuron culture requires different kinds of hormones, amino acids, etc. Now, in the brain, where they're actually growing, so these different factors are provided by glial cells. So we discussed about glial cells in the previous lecture, right? So all these factors are provided by glial cells in the brain. Okay. Now, but now when you are doing cell culture outside in vitro, then you will need something, some layer or something which can provide these specific factors. Okay. So this is basically a feeder layer. So this feeder layer of cells, what it will do, it will provide the same factors which the glial cells are providing in the brain for neuronal cells to grow. Okay. Now, if you just take glial cells and coat them in a dish, and then you think, okay, these glial cells will produce these all these factors, and then I can culture my neurons there. That is not so easy because if you go on and culturing these glial cells, they also produce some cytotoxic factors after a certain lifespan. So, in order to circumvent this problem, neurons are grown in serum free media, which was the neurobasal media about which we have already talked about. So, neurobasal media along with this B27 supplement, so that the neuronal culture can have all the important factors which it needs to grow, which the glial cells provide in vivo, and also it does not provide any cytotoxic effects. And then we have our neurobasal medium B27 supplement. Glutamax is also added, about which we discussed just now. So glutamax can also be added. So all these are going to now provide the important factors which are required for the growth of your neuronal cells. Okay. So next question. So this question is somewhat similar to what we discussed in the previous lecture. We discussed about xeno-free medium, right? So that xeno-free medium was also like, and xeno-free means animal-free medium. And this is also somewhat of the same thing. So animal origin free medium. So now the answer lies in the name itself. So you have to choose which of the statements are correct and which are not correct. So first is animal origin free medium is not manufactured using any raw material ingredients derived from an animal, including human, uh, from an animal tissue, cell or body fluid. No animal derived materials are used as starting material or in the primary level manufacturing process. So this is correct about animal origin free medium. Second is the components used to manufacture the raw material ingredients are not derived from an animal tissue, cell or body fluid. This is also correct because it has to be animal origin free, right? Third is ingredients and components may be produced by or extracted from plants, bacteria or yeast or cell-free systems derived from them, or may be produced by organic synthesis. So this is also correct. Then last is may contain recombinant animal or non-animal proteins provided they are expressed in plants or microorganisms. So all these options define or talk about what an animal origin free medium is. So here the correct answer will be all are correct, right? So I discussed last time like why is there a need to like not use animal origin components. So that is because as I mentioned last time, so basically we want to move from just a like research based approach to a translation approach where whatever we are doing in the laboratory can be translated directly to hospitals or to clinical trials. So that is why like now a lot of strategies are being used so that whatever cell culture media components are there, they are animal origin free. Okay, so that was all for today. Basically, all the lectures and stuff are uploaded on the YouTube. So I'll share the link in the chat box again. From there, you can watch the lectures again if you want, or if you missed any lecture, you can go to that channel. Which previous slide is it? Is it this slide? Okay, question number 10. So this is question number 10. So thank you all for attending the session. I'll upload this lecture on YouTube, so you can go through that.